Hi, teacher. Diam.
Hi guys, good afternoon.
Hello, sir.
Hi. So how's everyone? I hope you're doing good. I hope you're all healthy and safe. I've heard that in Batam it is now red zone again. So please be careful, everyone. Last time we were already green zone. Now it goes back to red. So please be careful, guys. Actually, whenever I go out, I kind of get uh, paranoid. Do you feel that when you go out? Even if you're wearing mask, no, it feels I feel paranoid at times. But anyway, so I hope you're doing all good. Um, for today, we will continue with what we have started last time. So we had a short recall, no, about your mitosis and mitosis and meiosis. For today, we will look at chromosomes. All right. I hope everyone is here already. Jesh, can you please, Joe and Jesh, can you?
Teacher,
Yeah. Yes, Aaron. What's the problem?
may I go to the toilet for a while?
Yeah, go ahead. Just go ahead. Go ahead. Okay.
Sure.
Oh,
Why?
okay.
Why? There is two teacher Crisel here.
Yeah, because I'm actually going to use a gadget I have just acquired, no, to be used during my class. So I hope I will work on this very well. So guys, uh, try to be patient with me today. Not try to bear with me today because I will be using another gadget that will help me in my class. So if it takes longer than usual, please bear for a while. All right? Maybe in 2 weeks or in 3 weeks, I will get used to this thing already. All right? So let's see. Let's see if it's effective or not. If not, I'm not really sure. This is costing me a lot actually. Yeah, but we will see. Now we will make use of what we have. All right. So, if we can just go back first to this drawing right here, to this illustration right here. You still remember this one? I think I showed you this last time, right? Yes.
Yes, sir.
Right. So before actually going to cell cycle and cell division, of course, we must first understand what is the exact structure that is actually involved in cell cycle and in cell division. And here and in here, we're actually talking about your chromosome. All right, your chromosome. Okay. So before we can actually move from chromosome to DNA, okay, we take a look first at what chromosome is. So like what I mentioned last time, your chromosomes are actually found in your nucleus, okay, in your nucleus right here. And inside the nucleus, there are lots of chromosome present. No? The number of chromosome depends on the organism. Now, in humans, we're talking about humans this time, how many chromosomes, let me recall our discussion last time. What, how many number of chromosome is present for a somatic cell or a body type of cell? How many chromosomes are we talking about?
46.
Okay, very good. 46, or we call it a diploid cell, meaning it has 46 chromosomes in the nucleus. All right. For how about for sex cells or your gametes? How many chromosomes?
23.
46. It's actually 23, or we call it haploid. I think you're
Deep.
familiar with that already. Okay. Are we good, guys? Can we follow? All right, so haploid and diploid, okay? Now, for today, before going to DNA itself, so remember that DNA is just a that your chromosome is just a long strand of DNA that is coiled together like this. So they are coiled, they are wrapped around, tightly packed like this, okay? So before we go to the DNA, let's go first to chromosome, all right? Let me go back. Okay, go back. Go back. Okay, guys. Okay, now here, let me just, let us try this one. Can you see this, guys? It's a whiteboard, right?
Yes, teacher.
Okay.
Yes.
How about this one?
Yes, teacher.
Yes?
Yes, sir.
Yes, teacher.
Okay. So, for today, we will look at chromosomes. All right? So, having observed what chromosomes look like, how can you describe it based from the illustration that I've showed you? Actually, guys, we're supposed to be looking at chromosomes under the microscope in the lab if we had class. No, we're supposed to be looking at onion cells, and we are actually going to see real chromosomes dividing, no, at different stages. We're supposed to be looking at that, but since we don't have time and you don't have microscope on your very house, so we will just settle with pictures for the meantime. But I hope we can still work on that. But anyway, going back to my question, how will you describe chromosomes? What do they look like? Anyone?
X.
You guys, just raise your hand if you want to answer. Yes, Clara, you mentioned about it looks like X, right?
X.
It looks like this one from outside, okay? Or maybe you're familiar with this drawing, yes? Right? Okay. I might be using this drawing, but I will not gonna be using this one. Okay? I will not use this one. I might be using this drawing, but if I'm going to give you the real, isn't it amazing? Can you see this, guys?
Yes.
Take a look at how I draw it. Okay? It might not look as what you can see from the internet or probably from the book, no? But this is your chromosome if we're going to look at it at the very close part. Okay? All right? Now, how are you going to describe your chromosome as I draw it? Anyone? So it doesn't just look like this, and it doesn't only look like this, right? It actually looks like this. So how are you going to describe me your chromosome? How do they look like? Or what do they actually look like? Anyone? Yes, Erica?
They look like springs
Mm-hmm. All right.
acting with each other.
Okay. So if they look like spring, actually your answer, Erica, I always use that particular analogy in describing chromosomes. If you're familiar with the spring toy that you used to play when you were little, it stretches, right? When you stretch it, it becomes longer, right? But when you congest it or when you put it back together, it only occupies a very small space. Am I correct? Are you familiar with the spring toy? So your chromosome actually looks like that, no? Now, if I'm going to describe it, all right. These are actually supercoil DNA, no? Or in other term, we call it condensed. Condensed. Okay. When you say condensed, it means it is coiled together very tightly. Or in other terms, you might have encountered this already in the books. They are tightly wrapped around together. All right. So these thing right here are your DNA strands, very long DNA strands that are coiled together, that are condensed together, and they are wrapped tightly around together. Okay? Hence, if you're going to stretch all the chromosomes in your body, okay, you're going to stretch it, all of it, all at the same time, it will reach up to the moon. That's how long it is. And what is the reason behind it being super coiled or being condensed? What do you think is the reason behind that? Alfred, what do you think is the reason why this DNA has to have itself coiled and tightly wrapped together? There's a reason behind that. What do you think? You can guess your answer.
so that it can fit in, so that all of it can fit in the cell.
Exactly the point. That's correct, Alfred. The reason why it needs to coil up so that it will not occupy as much space as it is already. So they are wrapped around, they are coiled together so that it will fit, especially inside your nucleus. All right, so I hope you're writing that down. I hope you're getting the idea. So again, your chromosomes are, I'm going to define what chromosomes are. These are cell structures that look like threads, or they are thread-like structures that carry all genetic material. Because remember, these are just coiled or condensed DNA together. Genetic material inherited, copied from gen generation to generation. Okay, right? That's how we're going to define your chromosomes. Again, chromosomes are cell structures that are actually found in the nucleus, okay, that are wrapped around together, that are condensed and super coiled, and they actually look like threads, no? When you look at it under the microscope or maybe from far. And they actually carry genetic materials that are actually inherited and passed on from parents to the offspring. That's your chromosome. Okay? So I hope you are familiar already with your chromosomes and the number. I think you're all familiar with that. Let's now go to the parts. Let me just erase this right here. Right? Now, let's go to the parts. No? Let's take a look at the parts. I believe, if I'm not mistaken, I have shared this already during SMP2, but maybe you don't remember anymore. Because remember, we have discussed, I think, your female and male reproductive system. So somehow I have touched already about this one. Okay? So let's take a look at the parts. Right? Take a look at the parts. Okay? Let's start with this two arm-looking structure on the upper side of your chromosome. This structure are called your P arm. All right. Now, in humans, usually, no, P arm is shorter. Okay. All right. That's P arm. So these two right here, these two right here are your P arms together. Okay. P arm. Now, this part right here, the lower part, these two arms on the lower part, is now called your Q arm. Your Q arm. Okay? And usually, this arm is longer than P arm. All right? Okay? So this is your P arm. This is your P arm. And this is your Q arm. Right? Now, together, they are held together by this structure found in the center of the chromosome called your centromere. All right? So the P arm and the Q arm is held together by centromere. Okay? All right. Now, in each of these arms, I'm going to use other, okay, let's try blue. Okay? All right. You may not see this in the picture or even under the microscope, but these chromosomes, on each of this arm, there is what we call telomere or telomeres. Telomere if it's one, telomeres if it's many. Okay? On each of this arm, on the tip of each of this arm, there is a structure called telomeres. It's actually found on the tip, on each of the arm. What do you think is this telomere for? What do you think is this for? Why is there telomere on each of the arm?
For protection,
For protection. For protection against what, Clara?
outside materials.
Material, okay? Remember that they are long, very long coiled strands of DNA. What happens if there is no telomere present on each arm? What will happen to the chromosome? They will be very unstable. Hence, telomere is for stability of the chromosome. Okay? That's what telomeres are for. Okay? So again, let's have a recap. This is your P arm, your Q arm, your centromere, and you have your telomeres at the tip of each arm. All right? You guys follow? I hope so. Now, before we actually conclude the parts, let me use colored green. I hope you can see the colored green part right here. At your centromere, at the center of the chromosomes, if you're going to take a look very closely on this structure, there are tiny hair-looking like structure on it. Can you see that? Whoa. Can you see the green part right here?
Yes.
Right? So there are, if I'm going to describe it, they look like hair. There are hair-looking like structures at the centromere, okay? But they are not hairs, guys. They only look like hair, how to say, strings, or just hair. But they are not hair, all right? Now these hair-looking like structures are actually called your kinetochore. Is that clear? Oh my goodness. My handwriting is bad. K. K. E. Yeah, I think that's better. Okay? These hair-looking like structures are called kinetochore. Now my question is, if you did your activity and studied about it, but not really, if you did not really study about it, if you have just observed about it, what do you think are these kinetochore for? What are their functions? They look like hair, right? Why do they look like hair, and what are these structures for? Anyone wants to try? Let me give you a clue. If you're going to look at your mitosis and meiosis stage, especially during metaphase and anaphase, I hope you can see that in your notebook. No? This kinetochore is very important during those two stages. During those two stages. What do you think are these kinetochore for? Anyone, guys, wants to try? Yeah, take a look at your drawing, guys. Take a look at your work. During metaphase and during anaphase, what happens to the chromosome? Usually during metaphase, if I'm going to describe it, it aligns at the center or the metaplate of your nucleus, right? It aligns at the center. Okay, and then after a short while, this metaphase, during this stage, after metaphase stage, which is anaphase stage, what happens to these chromosomes after they are aligned at the metaphase?
They are connected to strings.
Yeah, that's very good observation, Clara. These strings are actually called your spindle fibers. All right, take a look at this one. We call it spindle fiber or fibers. Now, these spindle fibers appear during metaphase stage. And during this stage, this spindle fiber tries to attach itself to the kinetochore. So the spindle fiber is attaching itself to the kinetochore. For what? Wait, yeah.
To separate.
To help separate them.
Who answer that?
First teacher.
Okay. Wait, yeah. I'm trying to, let me clear my drawing. Okay. All right. So, okay. During metaphase stage, these chromosomes align at the center, right? The centrioles are present, no? Centrioles are present at the opposite pole, no? On each pole, all right? Now, during anaphase, during metaphase, these spindle fiber are attached, no? They are attached, they appear from the centriole, okay? And then they attach itself to the kinetochore, no? They attach itself to the kinetochore of each of the chromosomes, right? Now, this spindle fiber is the one that is actually attached to the kinetochore itself. And then during anaphase, this chromosome split. No? They don't actually split, no? But they are pulled apart towards the opposite pole. Okay? So this spindle fiber attached to the kinetochore helps in pulling, no? It helps in pulling this part of the chromosome towards the pole, especially during anaphase stage. Right? I hope you're getting it. So those are what kinetochores are for. Okay? So I hope you can get that. I'm actually trying to print screen and put it in your line. Okay? So I hope we're good with this one. I hope we're good with this one. Any questions so far? Any questions so far?
No, teacher.
All right. I hope you understood it. Okay? Now, so we're done with the parts of the chromosome. Again, we have your P-arm, Q-arm, centromere, telomere. We also have your kinetochore. Now, let's take a look at these words. Let's take a look at these terms. Because I want you to differentiate these three terms. So we have chromosome, we have chromatid, and we have chromatin. Are you guys familiar with these three terms? This one maybe, yes. This one maybe also. How about this one? All right. Let me try to work on it in drawing. I'll draw it for you. Okay. Sorry guys. Erase. Erase. All right. If I'm going to draw a chromosome, I'm going to describe chromosome like this. Okay? That's chromosome. Now, what is chromatid this time? Maybe you have seen this when you were working on your activity. That's why I told you to be familiar now with these terms. When this chromosome splits and be pulled apart towards the opposite pole, the other half of this chromosome is now called chromatid. Okay? So this is your chromatid now. We call the chromosome chromatid this time. So this is your chromosome. half of the chromosome, which is this one, is now called chromatid. Now you will see from your book or maybe from the internet, you will see the term sister chromatid. So when you say sister chromatid, it's the other half of that chromosome. So they are sister, okay? Hence we call them sister chromatids. So during anaphase stage, later we will discuss that. The other half or the other sister chromatid is pulled towards the opposite pole, and the other sister chromatid is pulled together at the opposite pole, the other opposite pole. Hence we call them sister chromatid. Okay? How about chromatin? What is chromatin now? Anyone who wants to try? What do you
The
think?
one inside chromatid.
Is it the one inside chromatid? Again, remember guys, inside chromatid, you still have your DNA. How about chromatin? All right, let me give you a clue. If you have observed your drawing or your illustration during prophase stage, what do these DNA look like? What does the chromosome look like at this stage?
During what, teacher?
During prophase stage, at the beginning. Actually, even before prophase stage, during interphase stage. This is the first stage of prophase, at the prophase stage. How do these chromosomes look like? Anyone?
Squiggly. Squiggly looks squiggly. Like,
Thin thread.
Okay, they look like thread. Okay, if I'm going to draw it, it is going to look like this. Okay? So how will you describe it? How will you describe chromatin now? Chromatin are long threads of DNA that are uncoiled, and not condensed. All right? And this thing right here, when it coils like this, it is already becoming your chromosome. Okay? So, again, chromatins are long strands of DNA that are uncoiled and not condensed or uncondensed. So they look like long threads. Huh? Long. If you have yarn, are you familiar with yarn? Are you familiar with this?
Yes.
What happens when the yarn is not properly coiled? So it loses, right? I hope you got what I'm saying. Huh? When the yarn is not properly coiled together, or it's not well put up together, or maybe you just throw it out, it looks like that. It loses. It is loosening up. So it looks like this. Okay? So remember, guys, I have how many minutes left? Okay, I have 8 minutes left. Remember that chromosome, chromatid, and chromatin are all the same in terms of the structure. No? Whatever happens, hey, whatever happens, chromosomes, chromatid, and chromatin all contain DNA in it. These are only these are DNA. These are strands of DNA. What is the difference? The difference is not the structure, but its form. When you say form, this one. When you say form, they all look different, but they all contain the same. All right? So if I'm going to ask you later on, what is the difference between chromosome, chromatid, and chromatin? Teacher, they only differ in the form. They look different from each other. But they are all the same in terms of structure. Meaning, what they contain, it's still DNA. They only differ in the structure. So I hope you get what I said here. All right? These are all the same. They just differ depending on the stage that particular cell is undergoing. That's why probably when you were doing your activity, you notice that these chromosomes, these DNA strands, look different from one stage to another, right? But actually, they're all the same. All right? It only differs in the form. Okay? I hope that is clear. Oh, so how is it? How did you find this one? Is it effective, the one I'm doing?
Yes, teacher.
Because I think I will have a hard time just discussing this particular topic by just you guys looking at the PowerPoint. I cannot actually explain it just like this. All right? So I hope this one is effective. Any question, guys? Any questions so far? So for today, we are done with chromosome. I think you have understood what chromosomes are, what the parts of the chromosomes are, and even their different forms. No? Did I miss anything for chromosomes? I think that's it. Okay. Now, I will send a lecture on the different shapes and sizes of your chromosome because some chromosomes actually look like this. So this is the normal one. This is the normal one. Some look like this. Yeah. Okay. Some look like this, especially in some lower forms of organism. No? This is not found in humans. Not found in humans. Usually in humans, the chromosome look like this. This one you cannot find in human. But other forms, other organisms have chromosome that look like this. Some look like this, very, very short T-arm, okay? And others look like this, okay? So for me, this particular lesson subtopic is not very important anymore, but I will send the lecture anyway. All right? So let me pause. Let me stop my screen sharing already. All right, stop share. All right. Any questions so far, guys? That's your chromosome. Any question?
No.
So, next meeting, that will be next Friday, this Friday, no? I will move on from chromosome to DNA. So we will now discuss DNA, what it looks like, what is the structure, what does the structure look like, what is it composed of, okay? And then after that, we will move to cell cycle. That's where we're going to start in cell cycle and cell division. Okay? These are only prerequisite topics before we can actually move on to cell cycle and cell division. All right? So if there's no other question, I hope you understand that one. If you're having a hard time writing lectures during the class, I will try my best to put this in Word. I'll try to put my lecture in Word and send it to you guys. Okay? So if there's no question, I hope you find that fun. Although I'm having a hard time with my thing. I'm using this one, guys. So it's actually,
Tablet phone.
yeah, it's actually very, very, what to say?
Very helpful,
Yeah, very helpful.
convenient.
Because if I'm just going to use my mouse doing that, I don't think I can draw. It might look like monster or something, or alien maybe. All right, so thank you guys. I hope you had fun today and not confusing also. All right, I'll see you Friday. Bye guys, thank you.
Bye,
Bye.
thank you,
Good
teacher.
teacher.
Thank you, teacher.
Uh,
Bye,
Thank
teacher.
you.